I am more than excited to introduce our speakers today. Both of them are colleagues of mine here at Google. Josh Abramson, who's part of the Google DeepMind team, as well as Felix Schürmann, who is a senior HPC technologist and a very valued member of the Google HPC team here. And with that, Josh and Felix, thank you so much for joining our call today. And I'm going to hand it over to you to take us through the rest of this talk.
Thanks, Adnan. Okay, hi, I'm Josh. I work at Google DeepMind. I'm a research engineer there. And my background was actually in maths, not in computer science or biology. But in the last few years, I've ended up working on AlphaFold and related technologies here at Google DeepMind, and sometimes in partnership with Isomorphic Labs. So today, first I'm going to talk about AlphaFold 3, what it is, but I'll probably start by talking about AlphaFold 2, just reintroducing that because it might not be familiar to everyone. Then get into details of AlphaFold 3, look at some examples, talk about some of the successes, and then deal with some of the model limitations. And then Felix is going to talk about technical implementation, scaling, that kind of thing. Okay, so AlphaFold models. There's a kind of lineage of AlphaFold models. I haven't included AlphaFold 1 here, but the breakthrough model was AlphaFold 2. That predicts the structure of single protein chains. Then there was AF Multimer, which predicts the structure of multiple protein chains. So in the body and cells, proteins don't just form single chains in isolation. They group together into multimers where there's multiple chains kind of coming together to form a single structure. And then AlphaFold 3 deals with those multiple protein chains, but also all the other things that go on in cells, like DNA, RNA, and lots of kind of small molecules that interact with those biomolecules. So this will be explained in a lot more detail soon. So that's roughly the map. Well, why predict protein structure at all? And AlphaFold 3 predicts more than proteins. Let's just look at proteins for now. Predicting the protein structure from its amino acid sequence, so proteins are just sequences of basic chemical building blocks called amino acids, and you put those sequences together and then they fold according to physics and form some shape. So if you try and figure out what a protein's shape is through experimentation, it's a very expensive, long and difficult process. There's loads of steps. It can take a lot of investment and time, and every time you get to a new class of proteins, you have to kind of often start again with all the ideas of how to crystallize it and understand it. So it would be great if we could predict them, because then we'd be able to understand biology better, how it all fits together. Important number there is that about 200,000 protein structures have been determined so far. So that's not just protein structures. Each of those is like a complex of proteins and other things. AlphaFold 2 more or less solved this problem for single protein chains. It's got highly accurate prediction of the structures from the amino acid sequence. So you go from the sequence on the left to the predicted structure on the right. And in that arrow is contained the whole model. I think there's a slide later which has that a bit bigger, just to run through some of the highlights of what it is. So how does it work? It takes inputs, which are obviously the sequence that it needs to fold. Then a really important component is it looks up in a genetic database and finds all similar sequences that exist out there in the world. And that's really important because the conservation of certain correlations within that sequence tell you a lot about which pieces of the structure are bound together, because in evolution, if you break one side of that thing and not the other, well, either side, the structure breaks apart and it's not good. So if you find 2 points that are very correlated across nature, then that suggests they're in contact. There's a lot more to in MSAs than that, but that's the basics. We also look up similar structures from the data that's already been gathered through experiments across time and use that as input, but it's not an essential input. It's like an additional helpful bonus input. Then it goes into this complex model. So what happens? There's a series of neural networks that are all chained together. There's 2 levels of representation. One is at the sequence level. You have embeddings per amino acid that goes in, and then an embedding is just a vector per per amino acid. And then for each pair of amino acids, you want to think about the relationship between each pair, because you need to position them in space next to each other. You also have a vector and embedding that represents that, something about that relationship. And then there are special neural network processing algorithms that were created for AF2, particularly to deal with this pair representation, because you may have heard of attention, and in attention, classically, you just kind of attend to everything. So in the single sequence setting where you've just got a series of embeddings that's one-dimensional, fine. This embedding can attend to all the other embeddings and so on. As soon as you go into 2D, obviously that blows up, and it gets extremely expensive to scale the thing in the token dimension. So we came up with algorithms that did that in a more efficient way, only attending to sort of rows and columns in the way it needed to. Something famous called the triangle attention emerged, and that gave rise to the EVO format. EVO because it was processing the evolutionary data in the MSA. And that, in the red dotted area, is the kind of trunk of the model. It's the most expensive part. We rerun that several times to do the recycling. And then when that's done, it goes into this, this is AF2, a complicated structure module that uses some priors around how things should look in the amino acids, and then fits things together into a final structure. So in broad sweeps, that's the network of AF2. It also outputs these confidence metrics. So when it came to making something useful for people, confidence metrics are essential. And what they tell you is, for this prediction for the model, how much should you trust it? And they're kind of calibrated against the accuracy of the model. And this is really important. Not only how much should you trust this individual point in the structure, but how much should you trust the distance or the relationship between two areas in the structure. So there's 1D confidences are shown on the left and 2D confidences are shown on the right. And it's an essential component of AF2 that we definitely maintain into AF3. Okay, that's a very fast run through of AF2. I'm going to run through a couple of examples of things that don't immediately jump to mind when you think of protein folding. I think the famous, protein folding became very famous, and you think, okay, I've got the chain, I fold it. But this kind of multiple context is the first example of something that's a bit different that you might not have seen, where it's important to be able to predict the shape of a protein that you know all the other proteins that it kind of binds with in the cell, and then predict them all together, and then extract the shape of the individual protein you're interested in. So this shows this protein in blue there, binding with a bunch of other stuff. And when you predict it on the left with everything else, you get a better prediction of its shape than when you predict it by itself on the right. And that's why you had to move to predicting multimers even to understand single chains. In this example, it can really extend to large complexes. So these examples are from the CASP15 competition. That was one after the one that made AlphaFold 2 famous. And by tweaking the training of it to train on a bit larger structures, we managed to get a model that can be accurate on very large protein structures, thousands of amino acids going into these things with incredibly rich structures to them. And then we saw research in the outside world, don't know actually about then, this might have been concurrent or just before, people solved what we call the nuclear pore complex and other large complexes using AlphaFold to sort of understand pieces of it and fit it together. So it's not just, you're not just folding a protein chain now, you're combining it with experimental data and biological knowledge to create these huge structures that enable understanding of the cell. I realize I've said huge. It's still absolutely tiny, right? This thing is, you need a solid microscope to see it in protein terms. It's huge. Okay. Another important property is that you don't just run this model once if you want your best prediction. And that's important for high performance computing, right? You're going to run it hundreds, if not thousands of times for some examples, particularly antibodies. And then you use the confidence to determine what the best prediction is of those thousands of predictions. And you take the one with the highest confidence that's often got a better accuracy than all the other samples that you've seen. And we start to see this in more areas of using AlphaFold. That taking lots of samples was really important to increase accuracy on CASP15, for example. Okay, you can also start looking at, in the large scale, all proteins folded with all proteins. Let's restrict it to humans, so you've got 20,000 proteins and 20,000 proteins. Here's an example where they took 65,000 pairs of human proteins, and from those folds extracted a few thousand of the most confident, and said, okay, maybe these are genuine interactions in biology that we might not know about yet. And then there are experiments where you don't quite resolve the structure, but you just resolve the contacts between proteins. And they showed that indeed the model can find interactions that we didn't know about in biology so far. That's another situation where it's important to be able to scale your compute, because you're not just folding one protein, you're folding thousands and thousands of pairs. Okay, all this stuff was recognized by the Nobel Prize in 2024, Nobel Prize for Chemistry, given to Demis Hassabis and John Jumper for their amazing, groundbreaking leading work on AlphaFold. At the same time, it was given to David Baker for his work on protein design, and there's been a whole separate world of work on that that's also super exciting, also related to protein structure. Okay, that's AF2. AF3 comes in about a year ago now, in a paper published in Nature. This was work done together with Isomorphic Labs. And the reason why Isomorphic Labs are interested in this kind of work is because AF3 extends from proteins to proteins plus small molecules and a bunch of other stuff. But those small molecules in the body or in a cell, that can be a drug. And the binding of a drug with a protein, understanding that is key to pharma, quite clearly. So having models that can do those predictions for you in a way that's more accurate than simulation, faster than simulation, and enabling things that simulation couldn't do otherwise is really important. So to run through what it can do in a bit more detail, proteins. Obviously, we didn't drop the proteins from AlphaFold, but it also does RNA and DNA, and these small molecules, which are often drug-like things, but there's tons of other stuff going on in the body, glucose, for example. You've got modified residues and glycans. They're kind of special amino acids or bonded sugars, other technical things. The only things we basically didn't predict the structure of, and there are good reasons for this because of the structural data that's available, is where the water goes in these structures. And actually, waters can intermediate some interactions, and where the hydrogens go, because there's a lot of hydrogens compared to the other heavy atoms, and two, because the available data on where they go in the structure it's not widely available in an accurate way. But everything else is predicted, and you can sort of deduce the positions of hydrogen. Okay, overall, the model didn't change a great deal. So in this very simplified diagram, the inputs are the same, but now you've got more different types of sequences, and the outputs are the same. We predict some structure and with associated confidences. There were two major changes. One was that MSA processing was simplified, which made it much more efficient. Doesn't mean MSA isn't important, it's just an improved processing of it. And the structure module, that sort of complicated thing at the end after the trunk that generated the structure, that got changed into a diffusion model, which was a very popular model and still is a popular model for image generation, where you start with random noise and you gradually stepwise go to the final position of all your atoms. So each atom just randomly gets thrown in space, you step towards the final structure. That had lots of advantages. It's easier to train, it's easier to take all the different molecule types and then you just have to diffuse individual atoms rather than having a particular structure prior for each molecule type. That's roughly the model. There's lots of technical results here. I won't go into them. Basically, I will go into them a bit. These results show that across lots of different categories of biomolecular interactions, AF3 is state of the art. And in these examples, in these graphs, AF3 is going to be light blue. So perhaps the most important one is there's a breakthrough accuracy on protein ligand prediction. So when I've been saying small molecules, they're also called ligands. So think of these as the drug-like interactions with proteins. It shows that AF3 is significantly better than what's called this docking tool, AutoDock-Vena. And the docking tool for protein ligand interaction is simulation. It's using physics-based rules to take a protein structure and try and dock in some small molecule into that protein. You need to have the protein structure in place to do that. These tools only sort of simulate the movement or the position of the small molecule. They can be quite expensive to run, but there are much more efficient GPU versions available now. And there are lots of caveats about this graph. So since then, people have shown that you can do some amount better with docking tools, but those results themselves have a bunch of caveats. And I'll come back to that a bit later in the talk. Okay, so that's protein ligand. Then nucleic acids means RNA and DNA. So it's pretty good there, especially when you talk about protein DNA or protein RNA interactions. When it's RNA by itself, there isn't much training data available in the structural databases. And the model is not state of the art compared to other people combining models with human expertise and additional data. Covalent modifications like special amino acids, and then we didn't drop proteins. We made it even better than it was before at proteins, particularly for antibodies, so antibody-antigen interactions, which again has a sort of pharmaceutical relevance. We didn't drop the confidence. We showed that the confidence measures are correlated with accuracy. Roughly all of these things show accuracy on the right and on the x-axis and confidence on the y-axis, and that calibration is pretty strong. Zooming in on a couple of examples of these kind of protein small molecule things, just to sort of show you what it looks like in a cartoon form. We've got two different examples of proteins binding with drug-like molecules. And the important thing about these ones is, at least with the kind of available default docking tools, you couldn't get this successfully docked using simulation. So it's enabling things that just weren't accurate before. It's not like it was slow, we just couldn't do it accurately. So these sort of couple examples are the breakthroughs, and that's the setting. I said I'd talk more about comparison to docking tools. So you can get up toward this kind of scores that AF3 gets, but the direct comparisons are difficult. Firstly, as I said, you need the protein structure to dock against. And often in these studies, they take the ground truth, meaning the kind of solved experimental structure, and strip out the ligand. In that case, because it's taken from the protein already interacting with the ligand, the protein has modified a bit to fit the ligand. So when you redock it, it already knows something about how it's going to dock from the shape of the modified protein. If you just take the raw protein that would be without the ligand attached and then try and dock it, it's harder. There are other technicalities to their things, like taking the best of 5 rather than top ranked output. And while we know that docking tools give chemically more accurate things, like something I'll come back to later, there are also a bunch of mitigations for how to make AF3 more chemically accurate. Again, I'll show those later. I'm going to burn through some examples now, and I'm going to burn through them just because there's no need to sort of go into the biology of each of these. Partly I don't understand the biology of each of these, but yeah, I don't think that's as interesting as just sort of seeing the range of things that are being done with the model. So here's an example of a protein complex, and it binds with, you can see on the bottom right there, zinc and calcium. And it was predicted better with AF3 because of the zinc and the calcium, and increases our understanding of honeybees. Here's an example, another example of it interacting, the protein interacting with a ligand. So this isn't just us make a test set and showing, look, we can do better than docking. This is people out in the wild using it to develop understanding of the binding of a ligand with a protein. People are out there using it for protein DNA complexes, understanding these really pretty incredible looking interactions between proteins and DNA. Proteins have these wonderful interactions where they're chopping them, and then there are kinks in the DNA or the RNA, all sorts of things going on that are really surprising and difficult to predict. And then in biology, something that's quite hard to predict is the shape of membrane proteins. That's stuff that goes from inside to outside the cell wall and delivers stuff in and out. It turns out that you can kind of mock out a membrane in AF3 by putting in lots of fatty acids, which is lipids, which is what cell walls are made out of, and it does membrane predictions much better than AF2 did. You can predict the interactions of proteins with ions, and you can even start to use it to understand which individual amino acids are important for binding these metals by mutating the residues that are in contact with the metal and seeing if the confidence changes. So if the confidence gets much lower, then that's suggesting that this would break the binding. And we're getting towards the end of the examples. So you can use it to identify functional enzymes. So if you take an enzyme, which is a protein that catalyzes some chemical reaction, and you mutate it, then particularly when you're working with reactions that are new to nature, and this is a really exciting area of enzyme development, then the confidence of AF3 when you take the protein and the substrate that it's reacting with can be really informative about which mutations are going to help that enzyme be more reactive. You can even start to use it to model structural dynamics. So far we've just talked about predicting structure. That's not where it ends at all in the body, in cells. Things are incredibly dynamic. Enzymes are an example that I just had before of an incredibly dynamic process. The way DNA interacts with proteins is a dynamic process. The model doesn't give you a dynamic model, but you can set up all the different steps of a dynamic process and look at them and tweak things and try different versions of them until you find the most confident pathway. And then you can make some hypotheses around what the dynamic pathway of, in this case, nicking DNA might be. Okay. Right, I'm going to spend a bit of time talking about the model limitations. And there are these overall headline limitations. It's not a model of protein dynamics, and it can't model the physics of folding, and it just isn't always accurate. It is still ultimately a model. Often there are lots of questions around, oh, how are we going to solve protein dynamics, or what about folding? How does it work? The model doesn't really seek to answer those things with the training data they have or the way that it's trained. So I would like to highlight these first as things that we just don't really expect it to do. Like I said, there are things you might be able to derive by using static snapshots using this model, but the model won't extend to this kind of thing. Maybe I should talk a little bit more about this in the context of simulation, because there are molecular dynamic simulations that are supposed to be models of dynamics. And they're expensive to run. They get increasingly expensive to run as you increase the number of atoms. So when you get to proteins, that's quite a lot of atoms for molecular dynamics. And the levels of accuracy are generally low. To get to a high level of accuracy, the level of compute required is enormous, and in some cases just infeasible. Because if it was feasible, I think we'd see a lot more in drug discovery, people just solving things using molecular dynamics. But that's not the history we've seen, and AF3 has enabled much more than that. Okay, another limitation is that as you move away from the training set, performance drops. When you phrase it like that, it's not so surprising, but and we tried to do some analysis of this during the life cycle of development. And indeed, we saw that things that are less similar to training, we do less well at. A great paper came out analyzing AF3 and replica models externally, and that really helpfully showed this. So on these graphs, as you go to the left, we're saying that this protein ligand interaction is less and less similar to what we've seen in the training data, and the accuracy goes down. Importantly, there are two things. One is that AF3 is far above the kind of replica models, especially on the left, and that's the hard region. So yes, it's low percentages of success, but it's still taking it to a completely different level from where we were before, and at the moment from where the replica models are. Another important thing is that it shows that running more seeds and taking the top rank result improves the scores in that hard regime, in the regime where it's struggling, which is good because that means that sort of more compute can help. Okay, chemical violations. When you predict stuff in biochemistry, there are particular bond lengths, bond angles, and chirality, kind of the order of things, that have to be correct. And the model gets that mostly correct, except for chirality, which it really struggles with. But then you can use lots of samples and just take the one that's most accurate for the chirality that you care about. This is another slide on that. It's sort of saying bond length, angle, chirality, leaving atoms are a technicality we don't need to worry about here, but it's another chemical violation thing. Hallucinations is a fun one. So the model, we sort of took the term hallucinations from language models, and here it manifests in a very particular and manageable way. So maybe the word isn't quite right because it sounds scary, but what actually happens is there are regions of the protein which it should predict to be disordered. They don't fold into a structure, they just flap around. And often AF3 will predict them to have structure. And that's because when we train the model, we only train it on resolved regions that have structure. So all the diffusion head knows is how to produce things with structure. However, when it produces those regions, which should be disordered, it produces them in red. What red means is it's low confidence, and you should basically ignore it. So if you see a big red region, it's probably disordered and should be ignored. Now, we did fix this kind of using predictions from AF2, and so it's better than it was in the middle of training, but it can still happen. Here's an example of someone in a paper actually not looking at the confidences and thinking there was something there when actually the confidences show that it should just be ignored because it's a hallucination. We released this model in AlphaFold server, which has now had over 7 million folds go through it, and then later on GitHub for non-commercial use. But now there's GCP, so I'll hand over to Felix.
Thanks, Josh. Just before I really get into the GCP solution, I want to say I've been a computational scientist in my professional career, and I think AlphaFold really is a prime example of how computation that we've been doing through atomistic simulations has been revolutionized in the last couple of years. And today we're talking about the structure prediction of proteins, but I think that same revolution is happening in other areas, right? We've heard the talk about the WeatherNext models, and we're hearing other models coming out that sort of make predictions from the gene sequence, or sort of talk about make predictions whether certain structures are toxic or not. So I just sort of wanted to say that the whole method that we've been using of sort of simulating physics, I think, has gotten a new assistant and sometimes even a new master in the game. And I think that's really why we're having this series of the Google Cloud Advanced Computing community to talk about how AI is actually changing the very science that we've been doing. With that intro set, so yeah, I'm excited to talk about how we brought AlphaFold 3 to GCP, and specifically as a high throughput solution using Google Cloud and the cluster toolkit. Now, Josh explained it in a way, right? The input and the output, but I just wanted to here have a sort of a zoom in from a computational perspective. So the input sort of that goes into the model, which is this right block here, the model inference, has this traditional bioinformatics step of the multi-sequence alignment. And albeit this has been made more efficient with AlphaFold 3, it is still a hefty set of searches and computations. And that is done on CPUs and is data intensive. So essentially the search has to be done in parts through a number of complete databases. And the reason why I point this out is that if you want to go from the amino acid sequence to the structure prediction and the confidence scores, you have to evaluate the 2 steps. And one of them is an AI inference, and the other one is a bioinformatics step. And the AI inference runs great on GPUs, but the bioinformatics step requires CPUs and an efficient storage system. And so that's sort of when we looked at AlphaFold 3 from a cloud perspective, that's sort of what we wanted to map on cloud components so that you get an efficient execution of that model. So first of all, we started with the open source AlphaFold 3 model and container that our DeepMind colleagues released. And what the AlphaFold 3 high throughput solution allows you to do is to stand up an environment in Google Cloud, a project that uses this container and leverages the appropriate VM families, C3D for the data bioinformatics step, and accelerated VMs, whether it's NVIDIA L4s or AM100s or H100s. That's something we can talk about when it's relevant. That mostly depends on how large a complex you're actually going to want to fold. It handles the databases efficiently and scales out horizontally. That means you can fold 1 or you can fold 1,000, and it will just bring up enough data bioinformatics nodes, if you wish, these 3D nodes, to then actually fold these things in parallel, like a good high throughput solution should do. And it can auto-scale. So essentially you can have no compute resources on, and then as you're dropping sequences to fold, it can then allocate and grow dynamically this footprint in the cloud, and you can control how far it should grow. So whether you rather want to wait or whether you want to have all of this done in parallel. I want to point out the AlphaFold 3 model parameter terms of use apply. That's an important aspect, and we can come back to that at the very end of the presentation. But on the right-hand side, you see what that means. So essentially, the gray is Google Cloud, virtual private cloud. There's a Slurm cluster being set up, which has a logger node and controller node. Has then these auto-scaling worker partitions for doing the different steps, and they can scale up and down individually. And we have the storage to feed the databases and the input. So when we talk about a solution, here is the QR code, the short link. Really, it is a README extensive. It's telling you how to set this up. There's the infrastructure as code. So actually, we're using the cluster toolkit. I'll come to that in a second. That allows you to, with a few commands, take this infrastructure as code definition and then create your Google Cloud project out of that. And sort of batteries are included. You have the ability to log on to the logger node and then just submit folding jobs via Slurm commands. We provide some convenience functions there. You can activate a service daemon, which will just hover over a GCP cloud storage bucket. You can drop your sequence files in there, and it will just pick them up and process them. And we have currently in review upcoming there, the pull request is already out, a way to connect via the REST API via an IPython notebook to this Slurm server, and then sort of have immediate visualization of the results if you want to provide this to your users. So I mentioned the Cluster Toolkit. The Cluster Toolkit is an open source tool developed by Google that allows us to encapsulate best practices to set up certain types of environments, and specifically high performance environments. We use 3 different types of concepts. So the blueprints is the actual declarative definitions of an environment. Again, a virtual private cloud, a Slurm controller, for example, an auto-scaling, or it can be GKE cluster. It can be a batch environment. So that is something that you can do. So we have provided modules for the most relevant products in Google Cloud, and partners have also contributed. I'm talking about Slurm, right? In this case, SCAD would have worked together with us to provide a module that allows us to, whenever the user has in their declaration that they want a Slurm controller, that then the appropriate sort of setup steps are being done for you. When we're talking about setup steps, essentially the toolkit produces Terraform code. It can combine it with Packer to create, for example, certain VM images. And you can expect that. So think of it as we have a more abstract version than Terraform. The toolkit transforms this into Terraform, and then you can inspect the Terraform. And once you apply the Terraform, the respective cloud environment is being set up. And this is a very standard way for us to show our customers how they get performance, whether it is for aerodynamic simulations or large AI training. The cluster toolkit can set up the appropriate cloud resources for you. And so we've built one infrastructure as code definition for AlphaFold 3, and it is sort of focused on the high throughput. And so I wanted to speak about what does it mean to have high throughput and high performance. And so Josh already explained it, that essentially there's these two steps, unfortunately, are dependent on the sequence that you put in. So the length of the input sequence is the most determining factor when it comes to performance, meaning how long does it take to complete the data pipeline and the inference step. The longer it is, the longer it takes, and so the amino acid sequence and all the other complexes, right? If you have ligands or if you have glycans, if you have all of that, so that counts as additional input length, if you wish. So in the data pipeline step, we're using for the multi-sequence alignment the jackhammer, which is a reference application that is used for multi-sequence alignment. And in principle, it's linearly dependent on the product of the sequence length and the database size. So that just tells you the longer the input sequence, the longer it takes. And unfortunately, as it's doing an alignment and a search for certain types of sequences, it has more hits. And when it sort of enters these cases with longer hits, then the runtime can be much prolonged of that step. So in most cases, sort of it's quite predictable or determined, and in some cases it can take a lot longer, which is important, right? So in the solution, we talking about these two steps separately, the data science data pipeline step and the inference step, right? They can sort of use different amounts of resources and take longer. And so we now have a Schürmann controller that has two partitions, and we can essentially load the data pipeline step with jobs. And once they're done, we can inject that job into the inference step and don't need to run GPUs while the process is still in the data pipeline step. The inference step, on the other hand, is quadratically dependent on the number of tokens. So essentially the features that are being produced from the multi-sequence alignment, and therefore sort of also has a length dependent. And Josh also mentioned in some cases people want to have 1,000 folding predictions with different seeds to get different confidence intervals. Of course, then every time you ask for more seeds, that sort of the number of seeds, sort of you can multiply the runtime by the number of seeds, for example. And then one thing that can help. So on the one hand, you can use faster GPUs. But on the other hand, GPU memory is expensive, and you need memory size to accommodate certain sequence length. And there's a capability that AlphaFold can be configured to trade memory versus time. So if you do not have a GPU with enough memory, you can have the host copy memory between the host memory and the accelerator memory. And that sort of allows you to fold larger sequences, but it will sort of cost you additional runtime. So here is just sort of the solution in action. We have sampled 200 random amino acid sequences from the BigQuery dataset of AlphaFold, which is a pre-computed set of sequences hosted on Google Cloud. And here we stood up 3 of these C3D high-mem, which are AMD Genoa machines with a large memory. And in this case, we're using the NVIDIA L4 GPUs, which are in Google Cloud available on the G2 standard VM. And sort of here you see time, and on the y-axis is the number of active jobs. And sort of we've been dropping 200 sequences on the GCS bucket. and then you see sort of how Slurm will load the C3D VMs, and then sort of as over time as they the first ones complete and get out of this first stage, then so this last little hump are the inference jobs that are then scheduled to one of these 11 L4 GPUs, and sort of then it sort of goes down again because some of the data science steps, the data pipeline steps take longer, and then up again, and then so you have a long tail of things. And on the right hand side, this is a histogram of giving you runtimes of some of these jobs. So you see two distributions here, and the distribution with the longer runtime is the data pipeline step, so the multi-sequence alignment, which can have complete in 10 minutes or in half an hour, and some take longer, as we talked about before, depending on how essentially MSA is, the jackhammer is what it is finding. And the peak on the left-hand side, those are the inference steps, which typically run and complete in a couple of minutes if you have just a standard number of seeds. On this case, essentially on an NVIDIA L4. And here, the last thing I wanted to show is a little screencast. So here I'm on the GitHub repository of Cluster Toolkit. The links will be shown at the end. As I said, battery is included. So this is the daemon solution. You have just to switch on one switch in the config file. Here we are at the cloud console, looking at the GCS buckets. And what I'm doing is I'm just uploading here a couple of input files in the JSON format, either AlphaFold or AlphaFold 3 server version of the JSON file. See now they are arrived at the bucket. And here sort of I'm logged on to the Slurm login node. Nothing's happened, the queue is empty. Because the daemon is sort of watching over of that bucket with some interval. And then a few seconds later, you'll see that it has picked up all these jobs and scheduled them. And sort of if you have a large screen, you can see that they all landed on the same data pipeline node. So while we cannot compress the time of the data pipeline step, we can pack multiple subsequent alignment jobs on the same node. So Slurm takes care of that, and that's sort of what we mean with high throughput and efficiency, so that, I mean, we are scaling out the architecture as it's required by the number of jobs that you're using. With that, we're at the end. So there's a lot of links here, so we probably will stay on this slide to end. Important thing, we point out the license. So the open source model of AlphaFold 3 is for non-commercial use. So have a look at the license. But there's the AlphaFold server, which sort of Josh mentioned that is sort of free, and people can just log on and fold a certain number of sequences per day. There's, of course, there's the GitHub repository, and here you see that the last point, AlphaFold 3 high throughput solution on GCP with the cluster toolkit is what I just presented. The versions Josh talked about, and we have 2 email addresses to advertise. So if you are inquiry about AlphaFold 3, the scientists behind it, DeepMind are the right ones to address. If you're interested in running this solution or AlphaFold 3 on cloud, please use the cloud email. If you mix it up, don't worry, we talk to each other, and we also have your email to find the right addresses. And with that, Adnan, I think we're done and pretty much on time.
All right. Well, thank you very much, Felix. That was a wonderful session. We have a number of questions lined up, but I'm going to ask one which I think is very relevant to the content that you just presented of AlphaFold 3 on Google Cloud. And the question is from Bill Turner, and he wanted to know, what is the maximum memory available on each of the VMs?
So let me address, I mean, I'm not sure what exactly you want to ask. So the data pipeline steps sort of can make use of memory, but only up to a limited amount. So essentially anything between 16 to 64 gigabytes per job. And then essentially the databases are a couple of 100 gigabytes, which ideally are in an in-memory database as we are having it. So essentially, there we're heavily using the high memory version of our AMD Genoa platform. When it comes to the GPU part, we have anything, the L4s have 24 gigabytes of high bandwidth memory. We have A100s with 40 and 80 gigabytes, H100s with 100 gigabyte, and H200s with 140 gigabytes. And so that directly maps to sort of how long of an amino acid sequence you can map. So if you're interested in sort of, you say, which graphics cards do I need, contact us. We've run the measurements and can tell you which graphics card model you would use, or you can switch on the unified memory, and that helps you out when you don't have enough memory.
All right, thank you very much, Felix. All right, so the next question is from Joshua Williams, and Josh, I'm going to direct it towards you. And the question is, this is amazing work that you all are leading with significant enhanced vaccine designs, but also poses potential bio risks. How are DeepMind and Google Cloud implementing dual-use safeguards when it comes to providing access to models or computational resources?
The first thing I'd say is it's not necessarily for vaccine design or for design in particular. There are emerging examples of using AI3 and other structure-based models for design. The kind of safety aspects there relate to what's actually going to get produced. And so there's in the community discussions with producers of the screening. So there's already screening that happens when people are asked to turn DNA or manufacture DNA to make into proteins. And that's an ongoing discussion which we take very seriously. And if we move into more of a design space, we something we could take ownership of.
Okay, all right. Another question. This one's from Stephanie Linker. And the question again for you, Josh, is, are there any plans to tackle the dynamics of proteins and predict ensembles similar to BioEmu or Boltz-2?
So I wouldn't say that Boltz-2 has taken on the dynamics so much. You can run lots of seeds of all of these models, and that gives you a kind of an ensemble of predictions. Whether that prediction relates to the kind of Boltzmann ensemble, it's not clear. It might tell you all the different possible states, but maybe not the distribution over them, because all of these models are incredibly biased towards the distribution of states that you can create in crystal structures and put into the protein data bank, which isn't necessarily the same distribution of structures that you'd get for dynamics. So it is just hard to extend these models. One could imagine getting lots of molecular dynamics data if it was super accurate and kind of training on that. And that's a possible pathway for introducing more physics-based dynamics into these. But yeah, you can definitely take snapshots with all these models and try and understand all the different states. And the example with nicking DNA is an example of that, where you kind of take static points in this thing. And if you can find a high confidence pathway, then it's possible that's or you can make a hypothesis that that's the dynamics of the structure.
All right. Thank you, Josh. Okay, so another question, this one from Kiran Vasudevan. This one's for you, Felix, and it's a two-part question. And the first part is it possible for a pharma company to leverage AlphaFold 3 for commercial use?
The answer is yes, via isomorphic labs. So essentially, if you contact us, we will route you accordingly.
Okay, and the second part is, do we have a cluster toolkit version to provide the same implementation experience of AlphaFold 2?
You're not the first to ask. I think there's this highly asked for part, so stay tuned.
All right, let's see. Let's try to see if we have one more. We have. Okay, so a general question, and quickly, this one's for you, Josh. So what's next for AlphaFold 3? I mean, what's in the roadmap? Can you briefly tell us in a minute or so?
in less than a minute. I think a lot of that is down to isomorphic labs, because they're the kind of key consumers from my perspective at the moment of this model and where they want to take it. And I'm going to let them kind of drive that discussion. There's a ton of use going out into academia, and I think what's going to happen in the next couple of years is just seeing people run it with more seeds or more examples, and gradually it's just going to cause breakthroughs in understandings of structures that are more complex than just the protein-protein structures we've seen so far. And gradually over 5 to 10 years, and it'll be quite subtle, just an increase in understanding in biology and how it works. The model itself, you've got to think about what data sources are out there. The protein data bank is there. It's growing over time. There's going to be more RNA data. Again, 5 years from now, the rate of growth is not huge though. So there might be a gap for RNA where it takes some time before the model really takes a jump forward. And there's other data out there. So for example, Boltz-2 trained on binding affinity data. That's a very good source of data. It can be messy, it can be inaccurate, but there's lots of data out there.
All right. Well, thank you very much, Josh and Felix.